at which two different samples of DNA will denature. That means what? Become single strands instead of double strand. Now, sample 1 denatured at a lower temperature than sample 2, right? And researchers claim that DNA sample 2 has more guanine and cytosine.
B.
Can you just wait?
It didn't give us a second.
Shall I confirm or deny?
Wait, mister.
Whoever's listening to this is going to be bored. It's going to be faster, guys.
Okay, mister, confirm that. I didn't even read.
B as in boy, he's right.
So what makes C wrong?
What makes C wrong?
Because both of them have one.
It's not about single ring or double ring, it's about the hydrogen bonding.
Because A has double ring, the other one has single, the other one's also.
G with C has 3 hydrogen bond, A with T has only 2 hydrogen
So you
bonds.
talked about this last lesson.
I did?
No, it was either you or my actual teacher.
Your actual teacher?
So he's not the teacher?
No, like my school teacher I'm talking about.
So wait, they denature when they have a lot of hydrogen bonds?
No, they denature less if they have more hydrogen bonds. Okay, it's okay. Researchers studying the bacterium E. coli.
Just skip that.
Right, let's skip it. Sample 1 has a plasmid that has a gene that makes them resistant to kanamycin antibiotic. But sample 2, the plasmid of it doesn't have the resistance gene. Do you guys get what that means? Sample 1 should live, at least some of them will live if you give them the antibiotic kanamycin. But sample 2 will die because none of them have the resistance to this antibiotic. Okay. So look at plate 1. They only have nutrient. Almost all of them lived. Look at plate 2. They only have a nutrient. Almost all lived. Plate 3, nutrient with antibiotic. No one lived. No one lived. Plate 4, antibiotic. Only some lived. Why? Because those guys got the plasmid, those 7. And these guys don't have any plasmid. Good?
No.
Why?
Mister, this is so confusing. Why is there a long MCQ? I thought MCQs were simple, easy, short.
You thought wrong. There's like sometimes 5 MCQs based on one passage.
Yeah, I know. I
that's
saw
really
that
annoying.
yesterday.
It is super annoying, I agree. But we should never give up.
We don't give up, mister.
We don't give
We're
up.
aiming for that 6.
Okay.
I think it's D or it's B. One of them.
Which of the following is best supported by the data in table 1? Why do you guys think that plate 4, some of them lived?
Because it says 7. Oh, never mind.
Because they got the plasmid
Yes.
Yeah.
with the gene to fight off the antibiotics. So D.
And it says 7 observed, so obviously it's D. That's all I think.
Because of 7?
Yeah.
Wait. If it said 8.
If it says 8 observed, then it's.
What?
I don't know how to explain it, but in my mind I think differently.
If there's a weird number you think is correct,
Okay, guys. Q have ovules, so they're going to be female. And R has pollen, so male. Now, what percent of the genes, chloroplasts, will be inherited from plant R?
7.
What?
Plant R is a male.
Okay.
Can males give their chloroplast away, or only females could?
Only
All the
females.
females.
So 0%.
Zero.
Mister, what about something about mitochondria?
Same thing.
Okay.
It's
Only
zero-
females give it,
It's 0 because they're asking how much the male can give, right?
which is 0.
Oh.
Exactly. Oof. That's very long.
Yeah. So these are not MCQs.
Mister, how do we have 3 minutes for each MCQ if it's this much?
It's actually a minute and a half.
Wow.
High levels of, look,
That's what they expect us.
you guys should practice. The recording is not going to increase your Every time I get serious. Okay. High levels of a certain plant nutrient is runoff. It can lead to a lot of algae growth, which is called algal bloom in the aquatic ecosystems. Now, when the algae overgrow, they will also die in big numbers. And they will also break down or essentially decompose. And as they decompose, this will use up large amounts of oxygen in the water. And this will make oxygen levels low in the water. Therefore, aerobic respiration is not going to be supported. This whole thing is called eutrophication.
The whole process?
Yeah. Now, how could you tell how much algae there is in the water? By simply,
Looking at the graph.
yes, and measuring how much chlorophyll A there is in the water. Now, okay, so as we can see here, if there is more nitrogen, we also get more chlorophyll A. If there is more phosphorus, we also get more chlorophyll A. So both nitrogen and phosphorus are important for this organism, right? Which of the following best explains how higher concentration of nitrogen and phosphorus contribute to eutrophication? Let's see if you guys can think correctly. I said pre-solve, not post-solve, but anyways.
So is it B?
Mister, I was really
It's
confused
D.
because
It's
you guys
D.
already solved some last week, so I didn't know where to start. I solved the wrong pages.
Who said B?
I did.
Incorrect.
Yeah, no.
It's
D.
D as in dog. Exactly. 6. Which of the following was the dependent variable? Guys, why do you guys think it's D?
The colorful thing.
Mister, I think it's B because
I mean, sorry, do you guys know what 5 is D? Do you guys understand it now?
yeah.
To build macromolecules, right?
Yeah.
Okay.
Because when
You
I
said
studied, it said that this thing builds macromolecules.
6. Which of the following was the dependent variable?
A.
A. You just have to look at the y-axis. 7. Which of the following describes the relationship between the amount of chlorophyll A and the concentration of the nitrogen? If you guys look back at the figure, if there is more nitrogen, there's also going to be more chlorophyll A.
Yeah.
So they're
So you're just
directly
distracting me from my reading. B.
Yes. 8. Which of the following investigations would enable the researchers to test the claim that an increased concentration of algae has a negative effect on the aquatic invertebrates in the ecosystem? You're trying to investigate.
D. Mr. Fun fact, I wanted to be an
She's
investigator
right.
for such a long time.
And weren't you
Wait.
the one that said I want to be
Is it
like
the
a
C?
crime?
I don't
Yeah.
think it's
crime scene
D.
investigator?
I don't like
What
that.
happened? What do you
Sorry?
I don't get it.
There
Look.
isn't any jobs.
You know, yeah, you're right.
So I think
Look.
it's C.
It is
Who's
C.
like me?
Oh.
Yeah. Look, basically,
Why not D?
why not D? Chlorophyll A tells you how many algae we have. And the investigation is about algae. So focus on chlorophyll A. Okay? If there is more chlorophyll A, you're going to have more algae and you're going to have a negative impact on the invertebrates. That means more chlorophyll A, more algae and less invertebrates. So to investigate, and I think you said you want to become an investigator or you wanted to?
Yeah.
For what?
Crime scene.
Yeah.
Oh, really? That's cool.
But then I realized there isn't.
I don't think it's a female's job,
Yeah.
to be honest with you.
Not at least not here.
Yeah, only in the movies they like it's
Or
always.
like an interrogator, and like I ask about like what were you doing yesterday? Why did?
I think we're gonna get like here a lot of. Oh, okay. Every time we get serious, it's just. 9. Amylase is an enzyme that converts polymers into monomers by breaking a covalent bond.
By adding water.
By adding water. Thank you. Thank you. So adding water, right?
So it's either B or C. Because they both
B.
say in the addition of
B.
water.
B?
Yeah, B.
Yeah.
Not C.
How is it not C? Wait, if you remove water, you will build a bond, not break one.
Yeah.
Oh.
Which is why
Covalent.
C is wrong. If you remove a bond, water, you must build a bond.
Okay, nice.
11. Lactase is the enzyme needed to digest lactose. Most mammals produce lactase when they're young, but they stop once they stop nursing. And in humans,
Number 10.
sorry,
Why to skip number 10?
We go.
ahead of myself. Okay, question 10. Which of the following describes the most likely location of cholesterol?
Hey,
Can you wait?
Wait, I believe that.
Confirm or deny?
Wait, mister. Which of the following describes the most likely location of cholesterol in animal cell? Yeah,
Yeah, it's A.
Okay.
yeah, follow my path.
Now look, in humans, however, many people continue to produce lactase into adulthood and are referred to as lactase persistent. Okay. Which of the following mutations is most likely to cause lactase persistence? That they keep making lactase.
B.
A mutation that turns off the expression?
No, yeah.
You want to turn on the expression, right? Because they
C.
keep making it.
Mr. C.
So C.
Um,
They keep making it.
Mister, can you be there in the test for us?
I can be there as emotional support.
Sir, I'll bring my AirPods and put one on my left ear and call you.
Okay, I will not accept. No.
Mr. Close recording. I'm like, yeah, nice idea.
So look, wow. So cyclin is fluctuating. One time up, one time down. One time up, one time down, and so forth. But CDK is always there.
Constant.
Constant. So MPF is what? Is a cyclin CDK complex, which phosphorylates other proteins to start mitosis. The activity level of MPF depends on the relative concentration of the cyclin and CDK. Based on figure 1, which of the following describes the role of cyclin in the regulation of the cell cycle? Okay, go ahead, take a minute.
Can we all write the answers down?
It's better than saying it out loud,
Yeah,
yes.
Aiden. It's too quiet now.
You asked for that.
Okay, I have the answer.
Shall we say the answer?
Yeah.
Yeah,
B.
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 Exactly.
because cyclins are only in M phase, right?
Like they peak in M,
Yeah.
right? They peak in M, then decline afterwards. Yeah, you're right. Okay. If you have a triploid banana, it can't have kids.
Bananas can have kids.
How do they?
By the way, plants have kids, you know what I mean? They're called offspring. But anyways, moving on. There's a fungus that has been observed more frequently as a parasite in this triploid banana. And it might even go extinct. So let's explain which of the following could be the reason for this persistent fungus. Is that a cat outside or inside?
Yeah,
Sick.
so-
She sent me a video, she's like, your cat wants to get married.
What?
Yeah, I got the answer.
Are we gonna finish all this test?
What do you think? At this rate?
No. So we got like 10 minutes.
You guys should pre-solve if you want to finish faster.
I did.
I pre-solved the wrong test.
I said
Mr. Now
part
that
3.
I know about this, I'll start. And yeah.
I've been like discussing it for like ages now.
What is that?
Specific study
See.
3 tests from the blueprint a day, okay?
Look, sometimes if it's like short, yeah, you can do that. If it's like long, maybe like once maximum.
When will I finish? That's
You're gonna get
Start
a five.
today.
I can-
Start today. She's a grade 9.
Grade 9.
Just keep that in mind.
yeah.
Are you gonna see this up?
They always start in grade 9.
What's your name?
I don't know. Banner.
Lots of chance of failing, but yeah, some make it.
I struggle in grade 12.
Sorry, it's the truth, Yani. The stats say most of you guys fail ultimately.
Because the students in my school that were in 9th grade, they got like
5.
5 and 4, yeah.
Nice.
A lot of
I don't
them?
know why
Most
media,
of
they
them.
always get
Band
like
C, Band C too.
Leaks or no? Sorry.
لا لا ما اسمع.
Okay.
Promise.
All right, guys.
It's
So
gonna fail. No, mister, it's like the truth. What am I? He's in my class. Oh, it's fucking.
yeah, I don't think he's gonna listen to this
It
much.
was the only one that would actually make the lessons fun.
Yeah, I'm saying it was fun.
All right, so we have a type of squirrel called Belding's ground squirrel, and they live in close groups. And when they feed in the open, some of them watch for predators instead of eating. Ooh. And when the predator comes along, they give an alarm call to warn the others to run to safety. But this guard squirrel might then be attacked by the predator. It might even die. So why would they do that to themselves?
True.
D.
A.
Females?
Mom said there was like a there was something called, what was it?
What?
So you know there's like 3 things.
Mutualism.
I think it's-
Yeah. Like when you sacrifice yourself or something. That's what they do. And it's a woman, they're not gonna.
So you're gonna go with D because they already had kids, they won't have kids anymore?
Yeah, who cares about so the kids can get more kids?
But yeah, 'cause, man, they want the females to have more kids. Probably not. Not D,
Mister,
guys.
remember when you said Italians will go instinct like we want? Mister, isn't it?
I didn't say extinct, but like, it's not good for them.
Yeah, Mister, isn't it? C?
It's a.
It is a.
Oh.
Look, because they are close relatives to each other, so that means that, they will save each other. So, yes, his genes won't get passed, but the genes of his cousins, which are similar to his, will get passed. It's actually in the book. It's called altruism.
Yeah, that's what I was talking about, sir. That word.
How do you remember?
We studied this. You remember?
Yeah, I remember, but I have to
No, you-
actually record it.
Maria, we studied this in school.
I don't remember what
Blueprint.
Mister, just 2 more recordings. I swear, I'll be done.
Okay.
I can finish them in 1 day.
Look, guys. Look, I can no longer save you. That's it. I gave you the thing. Next week, you're like, what if I start today? No, that's
I
it.
have started, Mister.
The blueprint.
I will prove you wrong.
He is wrong.
ANT.
RNA don't have.
RNA don't have thymine.
Now you gotta think from
Right.
these.
Yeah. Yeah, how is this zero? Zero.
Do RNAs have thymine?
No.
No.
No, they have A, U,
Oh,
C,
yeah.
and
Oh,
G.
I didn't think about that.
Ow.
Yeah.
Is that good?
Yeah.
That's easy. That's nice.
Mm-hmm.
I like that, Mister.
When did the class start?
تقريبا
3:40.
لا ايش يسمونها تقريبا 45
45?
تقريبا.
So you finish what time?
15.
15?
Okay.
All right.
Trying to get out of here so bad.
Me, no, I actually have to go to a Baytar. Do you guys know what that is? Baytar? Baytar?
It's either for us or for pets.
No, dude, that's Baytari. No, Baytar, like the guy who like does like a move on you, like.
Oh, that's one that's gonna
Chiropractic?
attack your
Yeah,
bones.
I have like back and neck.
Wallah, I need it.
Mister, take over. We're all.
Come with me, wallah.
Oh, well.
I'll go after this. He's a bit crazy. And if he sees the ring, that's why I took it off. He goes crazy. He goes
Why?
nuts. He's like, oh no, it will do this and this and this and this, and you're gonna like, I don't know what. So I take it off before I go. And like the sun shouldn't be there.
Oh, Mister, the girls are gonna come hit up on you.
Dude, it's recording. Moving on. Okay.
Mister, there's this one bracelet. It like helps you balance. Is that true?
I have no idea.
Bro, what bracelet?
And some grandmas fall really easily, like they slip, but this bracelet,
Guys,
I swear, it's a thing.
Bro, they just lied to us. Just make-
look, melanoma is a cancer. Okay? It's a what? It's a cancer. And some cancerous melanocytes have developed resistance to the drugs used in the market. So there's a new drug called drug X, which is going to affect 4 different melanoma cell lines. We're just testing 2 of them. Okay. So guys, does drug X kill normal non-cancerous cells?
No.
No, because they stay flat at 100%, almost 100% survival. But does it kill melanocytes?
Yes.
It does.
What does it do?
It kills cancer cells.
Oh.
It kills cell line 1 and 2. You see how they're dropping? Which one is dropping faster?
The first
The
one.
100 one.
No, second.
The cell line 2 is dropping faster, correct?
How do you know it's called cell line 2?
Oh, that's it.
Oh.
Yeah. Very good. They also looked at oxygen, which is important. Why would someone look at oxygen consumption? It tells you if they're having aerobic respiration or not, right? Okay. You guys know what this sign means?
Blocks.
Block. And you guys know what this sign means?
Continue.
Activates. Activates. Okay.
Mister, you know what that reminds me of? It reminds me of the RNA polymerase when it's on the lagging strand.
Okay. Which of the following best describes the data in figure 1?
Oh.
That's figure 1, right? Yeah, that's figure 1.
Yeah.
What do you guys think?
The melanoma is the cancer cell, right?
Yeah.
It's B.
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 B is the correct answer.
So you're not good
Oh.
at FRQs or
I thought I said increases the survival. I read the first one.
Mr. Aren't I improving?
Look, I actually want to see an actual test taken by you at home
Okay.
without any outside influence, without a phone. Let me see the result and then we can assess you.
Okay. Which test do we do next week, Mister?
We'll continue this one.
We're doing that in school. Aiden, we're doing that in school.
Which
What is-
school?
Well, should we make it after iftar? I don't mind. Right now it's before. Oh, way before? Oh, that's like hard. Because we already booked one other. They never had this kind of book, by the way. One time it was 12. One time it was 5. One time it was 3. Every time it changed. One time it was, I think, 11. I don't know. Just depending on the day. Okay, question 17. Based on figure 2, which of the following best supports the claim that drug X inhibits oxygen consumption? Guys, do cells need oxygen? Probably do, right? So, if drug X inhibits oxygen consumption, then they should be dying more. Correct? Are they dying in C? Let's see. Uh-huh. They consume less oxygen when there's drug X. Let's see. Cell 3. So, you must always compare the cell to itself with the solvent versus with the drug and solvent. Okay? It's not gonna get you, don't worry. You will become a doctor, inshallah, right? You will see a lot of bad things. So, C. You see a lot of bad things, so this should be easy for you. Like dying people. Yeah. Dead bodies. Okay. What they like mess it up. Dissecting everything. Okay. So it seems that in the experiment we have less O2 consumed due to drug X. So the answer is exactly correct. 18. Based on the information presented, which of the following best explains why the researchers measured oxygen consumption as an indicator? Guys, O2 consumption is important. So you went inside the house? consumption indicates the. Tell me when. He's not. Okay. Indicates the well-being of the cell. Well, this is pre-solved. Okay, now O2 is what? What's the job of the O2 in the electron transport chain? Final electron acceptor. Okay. The final electron acceptor. So in the ETC, O2 is the final electron acceptor. So by accepting the electrons, the cells get to live. So the answer is Judy online? Oh. Oh, he won the first one. Still trying to find it. 17 till 19. So, no, it's all shit. I don't know. I don't know. Honestly. Never asked. We're getting in trouble now. Online people never talk. It's like a general thing. She joined the bisque group that day. I think that was her last time. Okay. Question. We did 17 to 18, right? Okay. Question 19. Okay. A researcher identified a compound that reverses the effect of drug X. So you can reverse the effect of drug X, right? Okay. So how would you reverse the effect of drug X? Drug X was the drug that they're investigating, and it's able to kill cancer cells. So drug X kills cancer cells. Okay. Good. Yeah. So which of the following shows you how we can reverse the effect of drug X? Well, drug X has to be there. Whatever it acts on has to be blocked. So you put it on so you must block the complex that drug X is working on to reverse the effect of it. Okay? So you know how it says drug X leads to the activation of complex V? That means activation. You must then undo it, which means you must now block compound B. So it's C. You must block the same compound that drug X is activating. This means block. I think it was told to us at some point. We did talk about it? So it is C. Good. I'm just going to read in the file. Did you ask me that last week? That one was about me starting that day. Okay. This time, it was smarter. So the hardest one so far was I'm done. What did I get this time? Well, so I think I got that's not good. Okay. My question was, in theory, it could be finish the papers. That's impossible. Well, how? That's good. Wait, is this 66? That's good. Is this 66? I don't know. Honestly. I can do 4 tests in one day. So just do one even check. Like if you're very stuck, but like not give up right away. And if it's a full test, never do that. Just take it like it is and suffer and get a 1. So what? Get a 1 now, just get a 5 in May. None of you have taken a single test by themselves, have you? Timed? Did I give you the blueprint? I said Princeton. I said blueprint. You haven't taken a test in last month? Okay, which of the following best explains how molecules like O2 and CO2 can move? Are they polar or non-polar? I even use ATP. That's not active transport. Or you just got the wrong answer. I said test 20 part 3. Yeah, it's part 3. Ions? They're not ions. So let's be. Guys, what is our dependent variable here? Dependent. CO2 fixation. Is it more for plant type 1? It's more for both soils, right? There's no overlap. Is there overlap? No. So it's more for plant type 1. Are they different in figure 2? Are they the same? Yeah. No difference. Why? Because of the overlap. Good. Which of the following statements about the rate of CO2 fixation and the two types of plants is supported by the data shown in the figures? Can you explain what's asking? Sure. No. So look, one time you grow the plants in a well-watered soil, which is the control, versus the dry soil, which is the gray one. I mean, sorry, the other treatment. Now, one time you expose it to 21% oxygen, and one time you expose it to 1% oxygen. That's 1%. That's what? 21%. It seems that the difference between them only arises when they're at 21% oxygen, but not at 1% oxygen. That's what it seems like, right? There's no overlap at this percentage, but there's overlap at the 1%. So having 21% oxygen results in a significant difference between the two plants. 1 and 2 in both the control and also the dry soil. Okay, so we can take out B because they're overlapping at the 1%. You can't say this one is higher and this one is lower. Wait, it's the plant type 2 has a higher, yeah, they're overlapping. And yeah, you are correct. Statistically, plant type 2 has the gray one has, yeah, he's right. He's right. This is smart way to do it. You should always use elimination. I didn't read it. I read the you always find one. That's good. No, no, no. There's like 2 that are very close. I think that are very wrong. I'm not gonna mention, by the way. In solidarity. Okay. So we have here phases of the cell, right? We have S phase, G1, G2, and M, correct? When you merge the 2 cells, 1 and 2, if one was in the S and one was in the G1, when they got fused, they both become S. Now, guys, don't forget, it's G1, then S, than G2. So that means that the G1 will be upgraded to the S. What? Yes. So now if you put S with G1, they both become S. If you put S with G2, they both remain like they are. Put G1 and G2, they both remain like they are. Put interface with M, they both become M. So it seems that M will make interface also M. And G1 is, sorry, S is going to make G1 S. So S can influence G1, and M phase can influence interface. That's it about the number of the chromosomes, because that does not change. Yes. D is not. There is no mention of mutations. C. Amount of genetic information change. I don't know about that. You are transitioning. The messenger is not shown, but something is making the cell change its phase. Maybe a hormone or a chemical messenger or some compound. It's probably going to be A. Okay. I think so. Okay. Okay. Here I see transcription and translation occurring at the same time, in the same place. So that means that we have a prokaryote. Right? And they're both occurring where? In the cytoplasm. Yes. You know how in eukaryotes, transcription happens in the nucleus, but then translation happens in cytoplasm? But here we see they're both occurring at the same time. Meaning what? It's a prokaryote. Yes, it is C. It's a prokaryote. Yes. Since we see RNA polymerase, that means we are observing transcription. Since we see ribosome, that means we're observing translation. And they're both occurring at the same time. 24. The scientist investigates the possibility that two traits are determined by genes that are on the same chromosome. Same chromosome reminds me of the word linkage. Linkage. The lack of crossover. It's the opposite. Let me just enter Zoom from my laptop because sometimes they struggle in Zoom. They get like stuck and stuff. Oh, I don't have internet here again. Yes. It was me done. What? This is not responding to me. The laptop is not responding. 20. Thank you. Let's see what Judy is saying. Okay, we're gonna be over soon. Yep. Ready? To see. Hold on. Who doesn't use ChatGPT? Start. That's a bit late. I have the paid version. I don't know anything. Okay, I'm just gonna join Judy's thinking so I can see what he's saying. Join. Yep. Okay. Oh, that's correct. So now, this is what Judy is saying. Yeah, why is it not the same? Why is it frozen? Judy, can you? Wait, it's frozen. Wait, what? Hmm? Okay. Was frozen all this time? You didn't say anything? It's your bad internet. I agree. But now we fixed Judy's problem. Okay. Let's find out the only person in the room. Look. Actually, there's no chi-square here. Like, there's no application of the... Exactly. Look. We have here 4 possibilities. Long stem, white flowers. Short stem, red flowers. Long stem, red flowers. Short stem, white flowers. What do we have here? 4 possibilities. Okay? So if you have 4 possibilities, what is the degree of the freedom? You take your possibilities and you subtract them by 1. So 3. That's the rule. Now, you should go to your chi-square, which is found in your reference sheet, which is this one. And when you have 3 degrees of freedom, and the p-value is 0.05. By the way, the p-value will always be 0.05 unless they say 0.01. You will get 7.82 as the critical value. That's one of the aspects of the chi-square, yeah. I'll push the recording. So the answer is going to be C. Good. The closest. That's the rule. I don't know. Get it? Let's do something here. It's pretty cool. Let's just see. I want to test something. Oh, yeah, it's working. Judy can see everything. Yeah, I can see what he's seeing right now. Oh shit! Sorry. It's frozen. I should not screen record as I. Okay, okay. Yeah, I should not screen record. Okay, got it. Sorry, guys. Or sorry, Judy. Hold up. Isn't Saho? I think so, yeah. Everyone says that. Alright, guys. Sugar gliders and northern flying squirrels are mammals that have many morphological similarities despite being genetically different. You know what this reminds me of? What? Wrong. Convergent. Lacus. Lacus. Show it. They never become one in convergent. Yes. They never become one. You can never make 2 species one. Okay. Now, they both live in trees. They both have flaps of skin. Okay. And they can glide from one tree to the other tree. But one lives in Australia and one lives in North America. So, how do they have those similar morphological? So it's not divergent. So there was one common ancestor. They live in different areas, so it's not sympatric. Live in the same areas. It's D. There's no mention of death, so it's not C. Yes, that's exactly correct. That's conversion evolution. Conversion. Okay. Which of the following best estimates the population size of the sea lions in 2000 based on the data shown in Figure 1? So that is possible to see your language. What? Just connect. Oh, shoot. That's like. No. Yep. Just connect the. Okay. I swear. About 15% of the exam is a giveaway. That's basically about like almost 8 questions are giveaway. Okay, 10 questions. 10 questions. I'll give away, like, you must get them right. Actually, I have the number. Should I check for you? Ready? You can see it too. No. It's 120. Look, divide 83 by 120. Let's see how much you get. Divide 83 by 120. No, but I got this from College Board. Downloaded it this year. So they have not changed it. That means it still applies. They have not changed it. Look at that. It sucks. So just get 70% you're good. Perfect. Yeah, he's a veteran. Like if they do it in grade 9, they get a 2 or a 3. In grade 10, 3 or 4 or 5. They better get that 5. If they don't, I need grade 12. Basic students and savvy students don't let them do it until grade 11 for some reason. 11 only calculus, right? Yeah, like since grade 1, like Tuesday AP. Yeah, I know. 27. The figure shows a phylogenetic tree of various members of the order Proboscidea. Thank you so much. Modern elephants. Which of the following claims best supported by the information? The figure Asian and the African elephants are the most closely related to each other. Asian and African? No, it's Asian and mammoth. Look. No, there's no logic group here because they just came from one thing. Anyways, look. So these two are very close to each other because they form like the triangle. That's why. So it's not A. Not with this. I think. The mammoth diverged from its most common ancestor with African elephants. Before the mastodon diverged. I should read the names or the scientist who finds it can name them anything they want. I think, Johnny. As a reward. I think there's like rules probably, but like, Sticodon and Paleomastodon. Anyways. Is this Sticodon a thing? Sorry. I don't know, but it died 2 million years ago. They died 2 million years ago, so there's no more Sticodon. Because here it says, like they stopped it 2 million years ago. That means they died. but the Africans still live till today. I mean, the elephants. So look, the common. Let's just look through B, C, D. B is complicated. I'll leave it till the end. Let's look at D. The common ancestor of the African elephant and the mastodon, African elephant, and the mastodon, which is this one, is the Paleos. It is true, yeah, it is. Because, like, we have to go back all the way to here to see where they come from. So, why did you make us? Why did you make us like, find out which one was the closest? Yeah. You cancel A. Oh, yeah. Let's just see at C. The mastodon and the stegodon are diverged from their common ancestor 2 million years ago. The mastodon, no, they diverged here. Which of the following is true? So it's not that one. Let's look at C. I mean B. The mammoth diverged from its most recent common ancestor, which is this one, with African elephants. Okay, so that one, basically. Before the mastodon diverged from its ancestor with stegodon. Mastodon and stegodon. It diverged after, not before. It diverged 5 million years ago, let's say. This one did like, maybe 20 million years ago. So it diverged after, not before. So it has to be D. Which of the following describes the most direct effect of a mutation in the DNA that encodes a cell's rRNA? rRNA is important for making ribosomes. Look for ribosomes. Look for ribosomes. It's, let's see. You said B? Transcribe RNA, that's going to be RNA polymerase. Yeah. So what makes D wrong? What's mixed? What? D? Yeah. Oh, that's rough endoplasmic reticulum. I'm sorry, Golgi apparatus. This is the job of the Golgi apparatus. Yeah. I know it's like so you asked me. Yeah, we're saying that we have a question. Okay. This is a very weird pedigree. Yeah. Yeah, this is weird. Okay. So first of all, is it dominant or recessive? It's no, they will never do half. Look, I know it's not recessive because there's a lot of affected, number one. Number two, recessive means, like, let's say these guys are sick, but their parents are not. But here, if the parent is sick, the kids are sick. The parents are sick, and so on. So everyone gets sick. Almost everyone gets sick. It has to be dominant. There's 2 that are sick parents. How many sick females do we have? 1, 2, 3. 11 sick females. How many sick males? Oh. Yeah. Definitely. Definitely. It's excellently dominant in that case. Yes. Excellent. If it's dominant, appears mostly in the females. If it's recessive, appears mostly in the males. Okay. So, which of the following is the genotype of the individual identified with asterisk in the pedigree? It's a female, right? And she's sick. But her mother is not sick. So her mother was like, let's say, small c, small c. What do you mean? No, she got. No, If it's X-linked dominant, you must only get one capital C and you will get sick. Her father is like X capital C Y. So she became X capital C X small c. So she has one dominant and one recessive. Okay. I think you guys don't get this. Say something so I can know. Okay. Is the mother sick? Like the mother of the esoteric. Okay, she's not sick. And we said that we have X-linked dominant. That means if you're not sick, you must not have a single dominant allele. Yes. No, in the mother. The white one, let's say. Yeah. One outcome. He has to have the capital C. No, he only has one X. Males only have one X. And now let's make them X. Exactly. X small c, X capital C, Y. We get X capital C, Y, X capital C, X small c. So the female has to be this, the male has to be that. The female has to have one dominant, one recessive. So X and Y doesn't need either. Sorry? X and Y, even the math doesn't need either. Wait, how about if the female has two small? She's not sick then. That's the mother. Don't worry about that. I mean, these guys get sick by sunlight. What? They're very sensitive to sunlight. That's not good. That's not good. Really? He's right. Okay. Which of the following best describes the inheritance pattern illustrated in figure 1? X-linked dominance. Yep. You do. But in this case, they don't have it, so just don't look at it. Okay, 31. What mutation in, look, if it's italicized, that means it's a gene. Italicized, like tilted. Yeah. What mutation in this gene is the 4 nucleotide deletion? That's called a frame shifting mutation. You know what that means? Whatever comes after it will be destroyed. Is it going to be the... Sorry? No, that's different. That's for mating. So you might... How could you get a shortened protein? By getting a stop codon ahead of time. Ahead of time. So the answer is B. How quiet is this group compared to that group you joined on Wednesday? We spent 10% studying, 90% saying, please stop talking. And it takes twice a week. Okay, 32. Yeah. Oh, the one day they asked for a 2-hour lesson, they all, like, no one showed up except for last and second. Yeah, he's the host. He has to be there. Others just dipped. 32. A researcher claims that an individual has the disease based on a physical exam. How could we confirm the researcher's claim? How could you make sure that the person is sick? Using biology, by DNA sequencing. Because if you know the sequence of the allele you know it, like let's say you know it somewhere stored, if you sequence that person's DNA, and if you see the same sequence of that bad allele, then you know that it's a mutation. So let's see. Okay. Next week. Mister, what if I start today? Look, one serious question. From the blueprint files, how many have you ticked? Only three? Three in a week? We're not gonna finish. How about you? Go ahead. You put it for us. I did divide the number by the weeks that you have. No. Count from now. How many do you have? Yeah. At some point, it's like May 1st. Mister, if I start today, that in 4 days. Like 6 a day or something. Okay. Which of the following is the evidence that eukaryotes and prokaryotes share a common ancestor? I don't see it here though. Endosymbiosis? But it's not here. Prokaryotes do binary fission, not mitosis. Yes. All actually some prokaryotes use inorganic called H2S. This is the second one. The heredity one is the most annoying one. Yeah, also lots of detail. Lots of detail. Oh, okay. That's chapter 8. Yeah, mitosis. It's mental. So your weakness is his strength and vice versa. So everyone here just work on the same thing and get me those 5s, okay? Give me those 5s. 5 times 5, I wanted 25 in May. 25. Oh, and Judy, 30. Okay. Wolves, once native to Yellowstone National Park, were hunted to the point of complete extinction. That's lots of hunting. Now, as a result, the elk flourished. You guys know what that is? It's like a big gazelle or something like that. But of course, they can overeat, and they will destroy the trees and destroy the beavers. Now, after many years, the wolves were reintroduced into that same park. And by having them reintroduced, the beavers returned to their good well-being, the birds, and so on and so forth. You know what that means? The wolves were a keystone species. With only tiny numbers, they make the entire ecosystem balanced because they eat some of the herbivores so that they don't overeat their own food. So, this is ecology. Okay. Is right? Oof. Again, this sign. This means what? Block. Epinephrine is blocking the beta cells. And this means activate. Right, so epinephrine is going to block insulin production, but epinephrine is actually going to stimulate glucagon production. Okay. Epinephrine is a chemical often released during periods of exercise. Inhibit means stops it. Yeah, like blocks it. Epinephrine activates alpha cells, causing the release. And yeah, exactly. Nice. That was quick. Yeah. It is insane. I never said that. How did I call him dumb by saying you got it right? No, but the answer was so clear. The box down is no, see, some questions don't require background knowledge. Just common sense in the question itself. Like, you must never panic. You must never panic. And stop studying the book and the Khaleelcheats and the recordings. Pesticides are chemicals that are often sprayed on crops to kill plant-eating insects. Pesticides are effective initially, but then at some point, the insects become used to them. Okay? Which of the following best supports the claim by scientists that the pesticides will eventually lose? Yeah. He's right. Oh, wait, sorry. It's wrong. Look, saying they're going to build up tolerance means that they will choose to evolve. But you cannot choose to evolve. No, there's no tolerance by getting physiology. It builds up by itself. This is wrong. Look, what he's saying is over time they just become, more resistant. Which is wrong. No, some of them have a mutation that makes them resistant. And they will live and they will give rise to kids who have the same exact ability as them. So it's B. Yeah, because they're born with a mutation by nature. Yeah. Like some people are born to be, let's say, a nerd in something. Maybe it's computer, maybe it's music. They're just born like this. You cannot change them. Like you can be born to be intelligent, but you might waste it on something like crime. Yeah. There's like some very smart killers or like drugs. Okay, before they get caught. Okay, 37. I mean, Jeffrey Dahmer was very smart in cooking human meat, but Netflix. It's the best. I'm studying more than you are, probably. I slept at 3 a.m. I woke up at 8 a.m. Prepping for lessons today, all practice. 4 subjects. Okay, I'm just being a student. It's easier. Certain proteins in the bat. Now, bat is not a bat animal. It's a brown adipose fat tissue. Weird, right? They increase the permeability of the inner mitochondrial membrane. Okay. Whenever they say, take it as a rule, whenever they say increase the permeability, it's a bad thing. It's a bad thing. It's a bad thing. It will disrupt the balance of things. You must always be semi-permeable. It's a bad thing. It will disrupt the balance. So look for bad things in the answer. Wait, slow. No freezing. Achievement. Look for bad things in the answer. Will you get more ATP? That's a good thing. Pick a bad thing. That's a bad thing. B is a good thing. Don't pick it. I mean, more ATP is a good thing. So don't pick it. Right. Let's just look at the thingy. No. Okay. Look. How can you uncouple electron transport chain and oxidative phosphorylation? How could you do that? No, you cannot do that. We're focusing on the H plus, which means the pH. Protons means pH. So, are you sure? Are you sure, actually? Well, now it's been like twice, but I believe you all the time. I fall for it too. Yeah, the answer is C. I chose B. I also chose B, C. Great minds think alike. I think you asked me before. I told you, given my age difference between you. Oh no, never. Like in one sitting, I can't because I've already seen all the tests. Like with the students, like it's gonna be like rigged, you know. It's gonna be rigged. It's not fair because I've already seen the test before. You don't get a 5. No, I'm positive because I've already seen the test before and I'll probably get a 5. If they do new job numbers, but they don't. If they do, I will. No, but there's a percentage. Yeah. Just get 70%. That's it. You get a 5. Same thing, still a 5. Oh, it's not fair, right? Listen up. This guy. He's not college board. I don't know if it's either one of them, but it's not college board affiliated. What? I'm feeling. Oh, here. Did I try that sponsor? No. Sorry, I'm still. Well, it's actually on the drive. Well, yeah, I did it. Okay. So let's go back and check out why B was wrong in the first place. Okay. So, oh, I understand why. Guys, look. This is the inner mitochondrial membrane. And this is the outer one. You see, I drew the inner one to be folded. Inner mitochondrial. More what? Yeah, more enzymatic. So, look, guys, the H plus has to move here. And now, after some point, it has to move back through only the ATP synthase. Only. But now, if we're going to increase the permeability of the inner mitochondrial membrane, what's going to happen is that they will simply flow back without the ATP synthase needed. That's not a good thing. So if they flow back without it, they're not going to accumulate here as much as before now. You're not going to get a steeper gradient. You will get a weaker gradient. So it's not B. And the answer is C. Exactly correct. Okay. So you will say. Saying what exactly correct? Actually, I think I do. In pea plants, flower color and the length of the flower's pollen grains are genetically determined. You know what that means? The environment is not responsible for them. We're studying homozygous dominant pea plants. Same, yeah. In this case, both capital with homozygous recessive pea plants. So of course, you will get hybrid in the F1. And now, but the researchers' data, right, that was recorded or observed was different from the expected. And they did the chi-square work, and they found the chi-square value to be 5.5. Okay, another chi-square question. Okay. Guys, if we are crossing the F1s with each other, and they're both heterozygous, what would you get? You would get how many possibilities? 4, because you will get 9, 3, 3, 1. So what's our degrees of the freedom? 4 minus 1, which is 3. Okay. Let's check. So if you have 3 you will get 0.05, and the answer is going to be 7.82 is our critical value. Now, since the chi-square value, which is the 5.5, is less than 7.82, we have to accept null hypothesis, which basically means we have to accept that all the observed differences are due to chance events. Okay? So due to chance. Please don't. What are you not going to skip then if it's equations? Yep. You're gonna freak out. Oh, B. A massive increase in the growth. Oh, I got scared for it. What? Where is it? Okay, wait. A massive increase in the growth of a specific species of algae resulted in record-breaking levels of potentially dangerous toxin being released into the water. Now, the researcher makes a hypothesis that this unusual growth of the algae was due to an increase in the water temperature. And now he wants to test his hypothesis. Which of the following is the dependent variable? Yeah, he's looking at their growth. Exactly. Yep. No, dependent means he's observing it. He's not controlling it. Yeah. Question 40. Which of the following is the most likely effect of a mutation in the gene coding for a DNA repair enzyme? Okay, so can you fix the DNA? No, because now the gene that does that is mutated. I'm having a hard time not trusting things. 2 times 9 80 wrong. See, alright? Oh. Yep. Right, we're gonna skip that for now. We'll go back to it if we need, because what part to read? I mean, there's like a lot of details. If NAF is added to the cells, yes. Look, MCQ is 50%, FRQ is 50%. In the MCQ, like I think 0.8 points. I mean, divide 50 by 60, I think 0.8. 20. Oh, yeah. You know what? Why are you even asking? Don't skip any, okay? You have 90 minutes. We have MCQ. It's not enough, right? 1 minute and a half. Divide 90 by 60. But some questions must take like only 20 seconds. Figures. That's why you should start practicing. Stop. Look, if next weekend we have the same conversation, please let's not have it anymore. This conversation. What if I start today? Only with me, bro. How about alone? It's like going to the gym only with the coach, and the coach is not there for 6 days a week. You like what happens then? So please, okay? Just take steroids at that point, right? Okay, NAF inhibits this step. We. No, I skipped the graph to see what they ask about it. Back to the graph. So if NAF inhibits this step, can the 2-phosphoglycerate become 2-phosphoenolpyruvate? No. Yeah, you're right. You're right. You're right. So this one's going to accumulate. Based on figure 1, the net number of ATP molecules produced during glycolysis from the metabolism of a single glucose molecule. Net number. It's actually 2. But let's check. Here you lose 1 ATP. Here you lose 1 ATP. Here you gain 2 ATPs. And here you gain 2 ATPs. You lose 2, you gain 4. Net is a gain of 2. Mhm. Okay. An increase in the concentration of protons in the cytosol will most likely have which of the following effects on glycolysis? If you have more protons, what happens to the pH? Becomes less. Is that a good thing? Yes. Look, a pH change is never good. It's like having a temperature too high, for example. Which is not good. So what's the answer? Yep. Which of the following describes why a glucose transporter is needed? Isn't glucose a polar molecule? It's very polar. Lots of oxygen, lots of OH, lots of hydrogen bonding. So it's not A. And glucose goes from high to low. High to low. Okay? Okay. No, glucose is not charged. It's going from high to low, not low to high. Yep. Not active. Facilitated. Yes. Because we need a transporter. Like transporter protein. By the way, we don't study carrier in the AP. We took it back in chapter 2, or the second chapter basically. We need for glucose as a polar molecule, you must have channel proteins or transporter proteins. Same. That was a long time ago. That was back in like I think September we took that. Right now it's March, mid-March. And so don't study chapter 2 now in mid-March. Stop avoiding your future. That's everyone's concern, and that's why he was supposed to take care of the chapters back in September and December and November and October. Not now. Are we gonna have who's gonna start the blueprint today? Me. Next week you will not say. What do you mean? Why didn't you ask? What were you guys doing since September? They only have one. Honestly, you have TS2. I think you have to register. Don't you? So you only have one. So you only have one. To fail, if they don't study. Yeah. Look, guys, you get get more experience. Failing is not an experience, by the way. It's like saying I broke my arm once, let me break it again. Same thing. No, but then you just don't break it in the first place. No, how not break it again. You don't break it. And that's why you know how you make mistakes. You learn from mistakes. But the thing is, getting those things. You just you wake up with the arm broken. What do you learn now? What do you do? Okay. Wow. Actually, I don't smoke at all. I must take my strepsils soon. Okay. What? You just have to look for the goal. What's your goal in life? Well, that's why you don't have motivation. Okay, anyway, so guys, if it's italicized, it's what? It's a gene. Italicized. Is it called cursive? Yes. Okay. Now, we have a problem with this gene that makes an enzyme that's important for glycolysis. Oh, she's right. It's actually an important enzyme for glycolysis. We did PFK, we also call it PFK. So based on figure 1, there's a figure. Oh, the same question. We're still on the same long question. Yeah, I know, it sucks, but what can we do? No, you know why? Because I can put the same like this beginning in every single one of them. Yeah. You don't have to go back. We're right next to it. Workshop follow best explains why a low carbohydrate diet is recommended. I mean, they can't even break down glucose, right? So that's why they must only have a low carbohydrate diet, obviously. So B, right? 46. A mutation in the upland cotton plant causes the development of chloroplast with a single outer membrane. Oh, is that a good thing not to have internal membranes? Exactly. Exactly correct. Okay. So are they going to function? Yes. No, they have no inner membrane. Everything happens with that membrane. Very big mutation, but they still function. Recording. Okay, now. Do chloroplasts move into within a cell? It's not. Okay. Do they take up CO2 with their inner membrane? No. Exactly. Do they make ATP and NADPH using their membrane? Yes. I made a face, but you guys still got straight. Come on, man. Not gonna be there, unfortunately. I tricked you by making a face, but it's the same face I was making for the past two wrong questions. Let me charge the laptop. Sorry, guys. As I charge, you guys solve this question, okay? Because we don't have something called pre-solving, I feel like, in this group. Okay, 40% of the group results. I said it's 20, 20. Sorry, guys. Like I said, please solve my talk. If you're talking, you guys solving? Why not? No. You solve now? I think you can solve now. Is that on? Okay. No, it's not. Scared. Okay. Now, what do we do here? Explain how. What was it called? Like a bunch of squares. We just put them diagonal. Nope. X capital X small times X capital X small. What's the chance of getting X small X small? What? 0. No, not 0. It's 1 over 4. Okay. So this is capital X. Same thing, yeah. Do this one. You get capital X, capital X, capital X, small x, capital X, small x. That is 1 over 4. No, we're not done yet. Now let's do it for the capital Y, capital Y, small y. Same thing. And now capital Z, small z, capital Z, small z. Same thing. Yeah, that's what I did. Okay. Mosquitoes are frequently a target of insect control strategies because of their ability to spread disease. One strategy is to introduce guppies, which is a freshwater fish, into areas where mosquitoes are a problem. Guppies can consume large numbers of mosquito larvae, which cuts down on the number of mosquitoes living to adulthood. Okay. But then there's a fear that if you were to introduce guppies, you might cause more problems. Why? Because they are able to tolerate a big range of environmental conditions. They also reproduce fast, just like invasive species, you know? Yeah, guppies, fish. Just like invasive species. So the answer is A. Exactly. This is this one. I'm not sure. I think 5. Yeah, with the heritage, with the mental thing. Yes. Increasing non-disjunction. That means they don't separate. Okay. So affecting the M stage. Because anaphase happens in M. So yeah, checkpoint 3. Right, checkpoint 3. We got it? Scientists studying wild population of mantled howler monkeys. Okay. So guys, we are having a birth rate of 0.22, but death rate of 0.12. There is more birth than death. Right now they're 13. Will they become more or less? More. There's more birth than death. So it's not decrease. It's an increase. Yeah, because it's not exponential, it's the same rate. Like, not too big of a difference between them. Also, exponential, if it doesn't the same rate. Oh, really? Yeah. V is the same rate. So V is the same rate. This is always 0.22. Always? That's what it says. Which of the following best explains how the prokaryotic expression of a metabolic protein can be regulated when the protein is already present at a high concentration? If it's already there at a high concentration, shall you make more of it? No. No. So you don't want to increase gene expression. You don't want to increase RNA polymerase binding. It's so A or D. Yes. You already have a lot of protein. Do you want to make more of it? No. A or D? Which one? A. Why not D? This is the same thing. Histones only exist in eukaryotes. This is modification. Yeah. Like, that was just changing fully, but that one just blocks it. No, that's not why. Look, there. This one is blocking. This one is preventing. Same thing. Same thing. But histones are only found in eukaryotes. This is the book, which you must have known before. Okay, 15. Why didn't you know that back in November, or October, or December, or February? What? Don't tell me. Don't tell me. Trypsin and pepsin. He's smart at wasting the class time. Trypsin and pepsin are enzymes that function in different areas of the digestive tract. Okay, they both have different pH levels, right? Pepsin likes a pH of 2. Trypsin likes a pH of 8. It's the peak. I'm looking at the peak. Trypsin is basic. Right, so it's not A. No, trypsin works in intestines. Yes. That was mentioned. Yep. Oof. Oof, oof, oof. Another one. Question 53 to 57. Started a bit late. Let's go there. What's not helping with the monitor, just saying, because it's hard to carry back. Almost entered your house. Almost. I knocked on the glass door, and I saw a woman eating on the kitchen table. I was like, no, that's not her. There's no one. I didn't see any shoes. I was like, where's everyone? Keeps their own house or something? Okay. Yeah, but I was like, where's everyone? I was confused. Okay. Wow. One group on starch, one group on maltose. Female's much bigger than a male. That's interesting. This is very interesting. What do they eat? Okay, population of fruit flies. What? So wait, if they grow on starch, they mate with each other more. If they both grow on starch. No, I think they mate and there's just more females that pop up. No, look. Starch with starch mate more than starch with maltose. Maltose mate more than maltose with starch. So that means they they mate based on the food type. Okay. Okay. Interesting. Moving on. Which of the following best represents the null hypothesis? You know what that means? There's no effect. You look for no effect. So females gonna start to change. So it's not B. Look, what is the idea of figure 1? Look at the caption, it's gonna tell you. Determine if the nutrient source affects mating. So you're gonna say, for null hypothesis, nutrient source does not affect mating. Just the opposite. Exactly correct. 54. Oh yeah, I said that again. For population of Hardy-Weinberg equilibrium, certain conditions must be met. Okay, what are they? You must have a large population size. Right, so it's not A. No migration. No mutations. No. Yeah. It's not required for this question. You will see in the blueprint. Just open the blueprint. I know what it is. He made prints for you guys. You haven't? So I know about that. He printed it last week for everyone. I've been extra. I've been like extra. Which of the following is best supported by data in tables 1 and 2? Well, what do you guys think? I think so. I think it's B. I think it's correct for us. I think it's exactly correct. Because look, starch with starch, maltose with maltose. That's behavior, and they might diverge at some point. Have a great day. Judy. And you as well.
So starch is a complex carbohydrate that is digested by the enzyme amylase. A mutation in the gene that encodes amylase prevents digestion of starch. So if they have a mutation for this enzyme, they have no ability to digest the starch. And if you're unable to digest the starch, that would be an autosomal recessive trait. And so in this population of 1,000 flies, 410 could not digest the starch. So what is this number in terms of the Hardy-Weinberg equation?
Recessive Q squared.
Q squared. She's right. So the Q squared is equal to 410 over the total, which is 1,000. So 0.41. So Q,
Radical that.
radical that is how much?
0.64.
0.64. Now we know the Q
So why did you do that?
to find because they're looking for the heterozygous. And heterozygous means what? Means 2pq. So now I found the Q. Now it's time to find the P.
P plus Q equals to 1. So
Yeah,
1 minus that, yeah.
0.36.
C.
Now they want 2 times P times Q. So you say C?
Yes.
Oh, okay. You did the math?
Yes.
Confusing?
Is it B?
No, it's not.
No, B is the P. But they want the heterozygous, which is 2 times P times Q. So 2 times P times Q.
Mister, when are we gonna get the chi-square question? Because the equation is very hard.
If it's in the blueprint, we'll see it. And we're going to do it. Okay. 57. Which of the following combinations of flies showed the highest number of mating instances?
Starch and starch.
Starch and starch gave you 22.
So it's really annoying when we have to like go back and
You don't in the online. It's going to be displayed on the left.
Mister, they haven't installed any like practice test with the online version.
They did like a small mini test.
Yeah.
Just test previews.
Yeah, but like it's 15 questions.
Yeah.
How do I have?
It just tells you, like, teaches you how to use it, the app.
Where do I find it?
Blueprint. Blue book. You gotta install the blue book. Wait, blueprint and blue book. I just realized that. Okay.
Don't they mean the same thing?
Maybe that's right. No. Well, at this point, you might start to think so, but because how many times I say it. But no, blue book is different.
How is oh.
It's
App?
an app, yeah. The same one used for the SAT.
مستر شادي يقول الشفقات.
Any other time? 11 to 1? How is 11 to 1? 12 to 1? 12 to 2? Alright guys, 58. The primary function of the kidney is to exchange molecules across the membrane.
Mister, what was 57?
Starch with starch. 58. The primary function of the kidney is to exchange molecules across the membrane between the blood and the urine. One type of the kidney cell has a basic rectangular shape, except for a single surface which is lined with tiny finger-like projections. Whenever they say finger-like projections, do you guys know what that means?
Villi?
Villi, which means more surface area.
Isn't it also flagella?
No. But just think about more surface area. So not the volume, but the surface area. Okay? Which is?
B.
B.
That was so easy.
Okay. What do we see here?
Electron.
Transport chain?
We see electron transport chain plus chemiosmosis. So we're seeing right now here something called oxidative,
Phosphorylation.
which is the last stage of aerobic respiration. On average, more ATP can be produced from NADH than FADH2. Based on figure 1, which of the following best explains the difference in ATP production between the two molecules? Let's look at NADH. Okay, and FADH2. So NADH starts at complex 1, goes to complex 2, and goes to complex 3, then complex 4. That's NADH. So NADH has to go through how many complexes?
4.
But FADH2 has to go through 1, 2, and 3 complexes. So FADH2 will go only through 3 complexes. So look for complexes if we have
B.
B. Which of the following best explains why ligase is required for DNA replication? Do you guys know the job of ligase?
Yeah.
Yes.
To connect the
Okazaki.
Okazaki fragments. So it's A, right?
Yes.
That was pretty easy.